Let me welcome the industrial biotechnology course. Now, after the antibiotics fermentation, let us go to the other area where biotechnology industry contribute a lot. But we know that single cell protein and how we define the single cell protein. Single cell protein is nothing but a microbial protein, as it has the tremendous potentiality in the market. Because why? Because your microorganism can grow very fast as compared to any other organism that we have, any other living system that we have, like plants and other things that we have. So, naturally that now question may arise that why we are not using this single cell protein to a great extent for human consumption. The answer to this question is that single cell protein, like when you consider the bacteria, if you look at bacteria contains lot of nucleic acid, and this nucleic acid causes some kind of kidney stone and gouts, and that is a major issue. But in case of animal, that is not a problem. So, animal can take it very easily, but we the human beings cannot do this. Still, we have some of the single cell protein is largely used for our in our day-to-day life. One such single cell protein is the Baker's yeast, and Baker's yeast used as a nutritional supplement in the bread making industry, particularly it is considered as the leavening agent. Leavening agent means that when we because the bread is prepared by preparing a dough with the help of that wheat, and when we prepare the dough, we add some kind of yeast powder in it, and then we keep the dough in the incubator for some time. When you keep the dough incubated for some time, then the bulging of the dough will take place. Because why the bulging of the dough take place? Because when you prepare the dough with yeast cells, that yeast cell will try to multiply. It is the living cells. When it try to multiply, it produce carbon dioxide, and since it is a dough, this carbon dioxide cannot go out, it remain inside the dough. So, your dough will be bulge, and then we take it out and put it in the oven for drying purpose. And that is why when you cut the this bread, we will find that lot of pore inside, and this is very interesting. And but a question comes, a question may arise that why we cannot use the sodium bicarbonate, because sodium bicarbonate also when you heat it will also produce carbon dioxide. But obviously that is used for some biscuit making, not for bread making purpose, because bread making we want the nutritional quality, because that the Baker's yeast contain about the 50% of protein, and not only that, it has also contains some kind of, it is a good source of vitamin B, and it slowly expands, because maybe when you heat it, the carbon dioxide may evaporate produce very fast, but when yeast grow, it grows slowly, and so the uniform structure of the pore structure in the bread that will be developed, and that is possible only due to yeast. That is why Baker's is largely used in different bread making industry, and bread is the common raw material, common breakfast item that we have in our day-to-day life. Let me go in details that the Baker's yeast is the common name for the strain of yeast commonly used as a leavening agent in the baking bread and bakery products. It converts the fermentable sugar present in the dough into carbon dioxide and ethanol. Not ethanol, it produces mostly carbon dioxide, little bit of ethanol, because since the inside condition is little bit of anaerobic, but not to a great extent, very small extent. The Baker's yeast is produced from the species the Saccharomyces cerevisiae, commonly used for alcohol fermentation process, is called brewer yeast. Now, yeast looks very nice under the microscope. It has the budding. So, I can show you, I have already told you before also, the yeast has a budding. This is called budding. The small budding that we have, it is a eukaryotic organism, is a source of vitamin, and it has high protein content, about the 50%, and generation time is 80 to 100 minutes. Now, if you look at the history of baker's yeast, it is a very old product, because if you look among different biochemical products, it is the very oldest product. The first report on baker's yeast was given by Mason in the year 1792. The baker's yeast was obtained as a byproduct in alcohol industry. Processes are improved in 1860, which is known as Vienna process, where the cereals were used to produce yeast cells. During World War II, the fishermen then developed the granulated activated dry yeast. I shall discuss about the activated dry yeast, because activated dry yeast can be preserved for longer period of time. German found that the cereal grains can replace the molasses present in the cane sugar, because when I discussed the alcohol fermentation process, then I told that molasses is the good raw materials for the yeast fermentation process. Why? Because it contains good amount of sugar. Also, it contains the vitamin B, which is very much required for the growth of the yeast cells. Now, particularly in Europe, the beet is used as a source of molasses, because in the European country they have the beet as a sweetening agent, and in our country we use the cane as a sweetening for the producing of sugar. Now, this yeast under absence, when in absence of oxygen, it ferments to produce alcohol, but in presence of oxygen it produces cell by budding process. This is the budding process, how the multiplication of the cells will take place. Now, this is very nice. You can see how the budding take place, and budding produces change of cells, how the cells they can bind with each other. You can see it. Now, types of baker's yeast. Baker's yeast are classified on the basis of the moisture content. Dry yeast are good choice for long, because that I discuss whenever we produce any kind of product, we have two type of products. So, one is called a short time, another is long time product. Short time product means that we says ready market, but as for example that mostly the baker's yeast is marketed in two different form. One is called compressed yeast, another is active dry yeast. Compressed yeast content about 70% of moisture content, and since it contains the 70% moisture content, so what happens that if you keep it at the room temperature, the bacteria that present in the atmosphere, that will grow very easily on the yeast cells, because it contains lot of vitamin, lot of nitrogen source and carbon sources. That is why it is to be marketed on the refrigerated conditions. So, the bacteria growth will be inhibited, but active dry yeast, the moisture content is about 4%. Under that condition, bacterial growth will be very minimum. So, we can preserve for longer period of time. So, depending on the long time and short time use, that we produce the two different type of yeast cells. So, if you find ready market, we go for compressed yeast production, because we have ready market, but if we have long time market, we go for active dry yeast production. The dry yeast forms are a good choice for long term storage, often lasting more than a year at room temperature. The types, the active dry yeast is consisted of coarse granules of yeast with live cells encapsulated in a thick jacketed dry dead cells and other growth media. It must first be dehydrated it can be stored at room temperature for a year or frozen for more than a decade. It is more sensitive than overform a thermal shock when actually used as in recipe. So, this looks like this. If we look at very clearly, it looks like pebbles. It is small pebbles. I shall explain more in detail. So, when you dry it, it forms kind of pebbles here. Now, in case of cream yeast, because this is a semi-solid mixture, is suspended the yeast cell in liquid and which can be siphoned and primarily used in the industry bakeries with specially high volume dispensing and mixing equipments. Now, deactivated yeast, the dead yeast which has no leavening value, used in pizza for pizza and pan bread dough. It is powerful reducing agent used to increase the extendability of a dough, and it is the source of vitamin, and it has the nutritional value. It looks like this, how this deactivated yeast looks like this. Now, rapid rise yeast, it is a variety of dry yeast that is smaller granules. It dissolves faster in dough, and it provides greater carbon dioxide output to allow the faster rising. So, this is rapid rising that is active dry yeast that we have. The instant yeast, it is the smaller granules that substantially that higher percentage of live cells per comparable unit volume. It is more perishable than active dry yeast. It also does not require dehydration. Instant is for a small amount of ascorbic acid as it is a preservative. It is marketed like this. But I told you that basically we have two type of yeast that is in the market, largely used in the market. One is active dry yeast, another is compressed yeast. Is the cream yeast with the most of the liquid removed, and because the moisture content is about 70%, it is soft solid and best known in the consumer form as small and foil wrapped cube of cake yeast. It is just like our butter cake. Butter cake, we know butter that half kilo that butter cake that we have. It is something similar to that. It can tolerate more tolerable at low temperature. So, it is like this. So, it is in this like butter we have this kind of form. This is available, but this is usually marketed under refrigerated conditions. Otherwise, this yeast will be spoiled. Now, let me talk about the production of this yeast, how the production take place. The maltose is very costly. Maltose, we know that it is a disaccharide. It contains 2 glucose unit. Starch and cellulose appears to be the cheaper, as for example, rice, maize, wheat, barley, potato and sawdust. The pre-treatment is essential to aid the release of sugar for both starchy and material and cellulose. And the enzymatic hydrolysis, that is amylase and acid hydrolysis, are used to pre-treat the starch. The cellulose is predominantly pre-treated by acid as to undergo complete hydrolysis with concentrated HCl at room temperature or dilute HCl at higher temperature and pressure. The molasses, the waste, it is not waste, it is a byproduct of the sugar industry. It is widely used as a raw materials for the yeast production process. So, apparently it looks like that starchy and this cellulosic material appears to be the cheaper, but only the problem is that we require to have some kind of pre-treatment before we can utilize this material for the growth of the yeast cells. But in case of the cane molasses, which is a byproduct of the sugar industry, so you can directly use for the growth of the yeast cells. So that is why the cane molasses is largely used by the industry. Now, raw materials that the molasses not only supplies the nitrogen, phosphorus, but also some vitamins such as inositol, biotin and pantothenic acid. The every vitamin has different purpose, because let me explain that. The biotin, because I told you that every cell has a life cycle like this. We have black phase, we have log phase, stationary phase and death phase. Now, this lag phase, this is considered as the lag phase. The lag phase, it will be extended if the media does not contain biotin. Biotin is here the biotin is a very important raw materials, that the biotin enhances the lag phase. Now, if the biotin enhances the lag phase, that means your time of fermentation increases to a great extent. But in absence of inositol, the organism relucts to ferment sugar. So, your organism will not ferment sugar. So, the media should contain inositol, and pantothenic acid, if it is not there, your organism will undergo the autolysis. Autolysis means the whatever sulfur protein, as for example methionine, that is a kind of sulfur protein, cysteine is a sulfur protein. So, this one degrade, it produces the sulfide. So, your biochemical character of the organism will be lost, your organism will be killed. By using the judicial mixture of, I shown you when I discussed the alcohol fermentation process, the best raw materials for Baker's fermentation process, if you mix the cane molasses and beet molasses in the 1:1 proportion. Because if you look at this vitamin, I was talking about the biotin, inositol and pantothiol, they present in different amount in beet molasses and cane molasses. Naturally, since they present in different amount, when you mix together, then you have significant amount of this vitamin present in the molasses. So, your growth of the organism will be very high, because that is usually recommended, that 1:1 proportion of cane molasses and beet molasses usually the best raw materials for the yeast fermentation process. Now, the yeast cells cannot grow without about vitamin. This is called BIOS. Now, in yeast growth, that concentration of sugar is 0.5 to 1.5%. If the media contains excess of sugar, and then it unutilized sugar break down to alcohol. Now, this excess sugar is known to act exponentially feed assimilation, and pH is 3.5 to 4.5, and by using ammonium sulphate and ammonium hydroxide buffer, nitrogen supplementation is done. Here again, let me point it out, I mentioned this in alcohol fermentation process. The ammonium sulphate is considered as the best nitrogen source, because the yeast fermentation usually carry out at the acidic pH. When ammonium sulphate dissolve in the water, automatically that is dissociated to ammonium hydroxide and sulfuric acid. Sulfuric acid is a stronger acid as compared to ammonium hydroxide. So, pH of the solution will go to the acidic side. So, you do not have to add the acid from outside to maintain the pH of the media acidic. So, that is why ammonium sulphate is found to be the best nitrogen source. Lactic acid bacteria is usually used in the yeast fermentation process. Now, this is very important, because when I discuss about the lactic acid fermentation process, then I mentioned the one important property of lactic acid is that it is a good preservative. And I have given the example that cheese production, that I will discuss the cheese afterwards, and the cheese is a kind of product, is the kind of process, cheese making is kind of process through which you can preserve the milk, protein and fat for longer period of time. Now, question comes that if when milk is the best food as per human beings is concerned, because the protein is the animal protein. We know animal protein, we have all the amino acids, they are well balanced. So, since all the amino acids are present, essential amino acids are there, so we can utilize that protein very easily. In the day-to-day life, as for example, we can take if we take the vegetables by same amount of protein, maybe so 1 or 2 essential amino acids are not present there, the utilization efficiency of the vegetable protein will be significantly low as compared to that of animal protein. So, that is why the milk, protein and fat is very good for human consumption. Now, we cannot preserve the milk unfortunately for longer period of time, because why? Because milk is a good media for the growth of pathogens, because if we keep it, then a lot of pathogenic organisms will grow there. So, this is a problem with this milk preservation. Even you do the pasteurization, we cannot preserve 3 to 4 days. After sometimes you have to discard it. But when you produce the cheese, that during cheese making process we remove the moisture from the milk and we concentrate that, and not only that, during that process it produces lactic acid, and this lactic acid acts as a preservative and so that it can preserve for longer period of time. So, like lactic acid bacteria in this Baker's yeast fermentation process, purposefully we inoculated with this with the inoculum, so that we can the storability of the yeast can be increased to a great extent. Now, lactic acid is used before the inoculation with the other organisms. Optimum temperature is 25 to 30 degrees centigrade. Now, growth requirement, because the first because I told you that during aerobic fermentation process, cells grow, and but anaerobic fermentation it produce mostly the alcohol. So, I told you that in case of aerobic fermentation process, the growth rate of the organism usually 10 times higher as compared to the anaerobic condition, because so amount of nutrient consumption in case of anaerobic fermentation process is much less as compared to the aerobic fermentation process. So, air is required in the aerobic fermentation process, because I told you the organism can take the oxygen which is dissolved in the media. They cannot take the oxygen which is prepared in the which is present in the air. So, air should be supplied to the media in the optimum amount. It help for the respiration of the cells. It prevents the alcohol formation, growth of the cells, agitation of the media, the removal of toxic product like carbon dioxide. We know when cell respire, it produces carbon dioxide, and this carbon dioxide is the toxic product. If you allow carbon dioxide to accumulate in the system, that will affect the growth of the organism. The surface area of the bubble should be large, because it contains large volume of air than the smaller. So, optimum rate of air is 1 ppm. 1 ppm means, I just explained that what do you mean by 1 ppm, 1 volume per volume of the media per minute. And foaming, to control the foaming, the anti-foam compounds like octadecanol, tetradecanol, hexadecanol or different oils are used to avoid the foams. So, different type of anti-foam oil we use also in this fermentation process. Now, stages, there are different stages we have for this Baker's yeast production. First is the selection of microorganism. You have to select the suitable, I told you the organism should be robust, we call it the industrial microorganism. It should, genetical characteristics of the organism should be same, genetically stable, or biochemical characters should be unaltered, and then selection of media for the development of seed yeast and the fermentation process, because this should be cheaper and this should use the and lot of different types of carbon source should be used, so that you have lot of choices. Then fermentation process, the kind of fermentation process also used, that is very important, because if it is start tank reactor is usually recommended, because your cells will be well suspended in the liquid, so that it freely interact with the substrate and multiplied properly. Then the separation of the yeast cells, that is also very important, that how you can easily separate the yeast cells from this and dewatering of the yeast cell. After that you have to remove the water content and packaging. And this I shall show you that one thing I want to highlight here, that suppose yeast cells, when you remove the water, then I showed you that kind of compressed is that it produces some kind of block. Now yeast cells are the segregated cells. If you want to, even you compress it and you want to produce some kind of block, then what will happen, as soon as you want to, it will break, because you know that the cells, they don't have the binding characteristics. So we add some kind of binder to it, like an agar, pectin, different type of binder that use, so that it can hold the cell together, and then and only then we can form the blocks. So this is in practice and distributed under the refrigerated conditions. So let me tell you that that whole process, how this compressed yeast and the active dry yeast, actually the production take place. That is the seed yeast we prepared, because this is the unicellular cells. So we can easily prepare the culture very nicely in the lab, R&D lab, and then we take this culture in the form of seed can. We take it to the inoculum and it's transferred in the inoculum vessel under aseptic conditions, and this is the aerobic fermentation process. We take molasses as a media, and then when you have, and this capacity of this should be 5 to 10% as compared to production fermenter, and then we transfer to the production fermenter. In the production fermenter we take molasses. Here the molasses, the raw materials that we transferred here. Then after the production is over, we cool that, and after that we pass it to the filter press. I shall show you that how the filter, I have already showed you, and previously I shall show you again how we use the filter press. Then we add some kind of binder, so that we can hold the cell together. We mixer and extruder. I shall show you the extruder. Extruder means we force the material to pass through the small orifice, and then we do the packing here, and we get the compressed yeast, and storage is 3 to 5 degrees centigrade. But when we produce the active dry yeast, we do we pass it through the dryer. We cut it in the small sizes to increase the surface area. So the rate of drying will be we know the rate of drying depends on the surface area. More surface area, more will be drying, and not only depends on temperature, surface also plays very important role. After drying, it contains about 4% of moisture. We do packaging mostly in the polythene package, and then we have the active dry. If we keep it in the cold, we can store for a couple of years. If we keep in the normal temperature, we can store for a year only. So this is how the compressed and active dry yeast can be produced. I shall tell more details in my next lecture. And what I try to tell you here, that what is the importance of the bakers is, because bakers is mostly used for the bread making industry, and it is used for the leavening purpose. It has several purposes, not only for leavening purpose. It is used for as a source of protein, as a source of vitamin, which increase the nutritional quality of the bread. And then this produce, we find the cane molasses and beet molasses 1:1 proportion appears to be the best for the production, is a good media for the growth of yeast cells, and time of fermentation is about 11 to 12 hours, and temperature is about 27 to 30 degrees centigrade. pH is about 4.5 to 5, so in the acidic pH. So since the size of the cells varies from 3 to 3 to 7 microns, so we can easily separate it with the help of plate and frame filter press. In the next lecture, I shall concentrate on the process in details. Thank you very much.